warm good morning to all of you. I welcome you all in day 3 of the workshop. So today, as the previous days, we'll be having 2 lectures and a tutorial. The lecture 1 will be on analysis of hydrolysates, and there will be case study of soya protein. Then we'll be having a tea break. And then the lecture 2, that is the lecture 6 of the workshop, will be on peptide characteristics and nutritional properties. And will be about the case study of whey protein isolate. And then we'll be having the lunch. After lunch, we'll be having the tutorial based on the other 2 lectures. So now I invite our foreign faculty, Dr. Manoj Kumar Rao, to kindly come on the dais and continue with lecture 5 of the workshop. So
before we start, I have a request with you that studies, if anybody have any queries, so they will note it down. And during maybe after the tutorials, we keep few minutes time to keep on discussing that. As I requested you for a startup, you are just tell our student about some plans which they can propose, especially in this area. Thank you, sir.
Thank you. Good morning, everybody. Let's move to our next session. Okay. So we're talking about analysis of hydrolysates, and one of the case studies we selected about is one of the grain proteins widely used in the industry and also extensively studied, but application varies from food to food. The case example, like we say, tofu, and some of the fermented product is tempeh or something. You all know this thing, and Japanese love their soy food. Without soy food, that diet is not complete. It's like in southern parts fermented products, and then we have wheat-based product here, some of the rice-based product in eastern side. So this is the dietary patterns, demography changing, and over a period of time, fermented product has gained popularity. So we are also taking fermented product here, like idli and dosa. So that's comparable to what people are eating. And the yogurt is also fermented product. We all know this thing. So if we look at the commercial practices and production, this graph suggested that in this is simple technology. We discussed about degree of hydrolysis changes with the concentration and time, and then the peptide characterization being done by using mass spec, and then your purification is one of the aspect of that. The purification can be chromatography. So routinely you are using like enzyme purification. You can use NMJ, iodine, or all types of chromatography you are using here. So the amino acid release is also mapped. So that is a key factor of the peptides produced. Coming to the soy protein. So we're talking about 11S glycine. That's the major storage protein of soy protein that's used in generation purpose. The 7S, the protein is different from the 11S fraction. 7S protein, the beta-glycine, they lack the divide of the SSB, supplementary amino acid. So they do not form gel, strong gel, with gel. Even though you buy that as an ingredient, your tofu will not be texture-wise perfect. So consumers will not accept it because same processing technology we are using here, same temperature, due to the structural difference from the existing protein. So that's the classic example how structure affects the functionality of a protein. It's a simple example, everyday practice. But coming to the purification protocol, like any seed, you know that's how the protein deposition takes place during seed maturation, starting from day 1 to the synthesis of protein. the accumulation of protein, then maturation of protein, and then the storage protein and fat inside the seed. That's why we eat this grain food. That's the grain industry. That's the cereal or anything legumes. Basically, soy protein is a legume. So cleaning and dehulling. This is the removing. You can use clean meal. You must be using in your lab or in your institute or universities. And removing the seed coat. It can be done mechanically. So once the seed coat has been removed, the fat extraction. So you can use NXN to take out the fat. It will interfere with the protein functionality. So the idea is to, if you're using a substrate extraction, you can also take out the fat. But if you're using a lab scale, you can use a chilled mortar and pestle, add some of these reagent, and then grind it, then put liquid nitrogen. So you can also remove the fat. To dry it, just go down. Then the defatted soy flakes are there. So there's a grinding here. Then aqueous alcohol extraction. So that's called the soy protein concentrate. Means the whole protein has been taken into the extraction. Do not confuse it with terminology with soy protein isolate, with soy protein concentrate, with different taxon of texturized soy protein. That's why industry has evolved in such a way. These are the ingredients having different functionality. So industry will not buy soy protein isolate for some formulation because they have to pay twice the price of soy protein concentrate. So like whey protein isolate, concentrate and whey protein isolate. So if you have aqueous extraction protocol here, so we can see soy protein isolate here. So the purification, this is a flow chart. So how do you extract, what temperature, what chemical, how the pH, everything. But this is a flow chart, just to give an idea. So the other people here, they have already given how to extract soy protein in the lab, glycerin. I think we all agree that it's a very simple protocol. The industry is using that protocol. Then soy fertilizer, then defective slow fat flour, and there's texturized soy. So what happens with texturized soy? There's extrusion here. Means one of the industrial application of is called texturized meat, something like meat analogs. So soy protein into extruder. So you can design this experiment using capillary rheometer. Capillary rheometer basically has control temperature, pressure, shear rate. Once you know your formulation, you design the experiment and pass through the capillary rheometer, apply the pressure. It's being cooked inside the when it's being passing through the extrusion process. So the example is complex. So you all know the complex. It's basically an unextruded product. So the soy protein is texturized meat. Texturized meat is a meat analog. It tastes like meat for vegetarian diet. So people are using this texturized meat here. So soy protein isolate has been purified in this format. So we have our ingredient is ready. I mean, it can be concentrate or isolate or texturized soy protein depending on the application and functionality. Is there any question in this, or I'll go to the next slide? Okay, thank you. Then we have flowchart for experimental characterization, designing the experiment. So we discussed about soy glycerin. This is a model protein. We decided that we'll discuss about some of the model proteins, some simpler things. The protein, every protein is complex, but we have, when you understand the properties, it will behave like a very simple molecule to you. So it's like that. It's a gadget actually. It's like a smartphone. The function it's a complex thing, but accepted in a simple way. All molecules are like that. So soy glycerin, the two different concentration, normally the enzyme that's being used. So once a hydrolysate is produced, we also discussed how the hydrolysate is being produced. So we know that what are the parameters, how we design experiment. Then we have to characterize. That you can do any of the characterization. Functional aspect you can study. You pick the soy protein hydrolase fraction, solubility testing, also apply the content. So how they are going to affect, you can compare it to the native protein with the concentrate or hydrolysate. Then you have the clear picture in front of you, how the hydrolysates being produced and then compared with the other proteins. The degree of hydrolysis having two protein concentration so yesterday we were talking about a similar rate of concentration. Even though we increased the bromelain concentration twice the amount, so we didn't see any significant difference. The idea is that if we achieve the end product at lower concentration, the idea we discussed, we are not going to put high concentration of enzyme into the system, so that will have detrimental effect. So we can lower the concentration and we are having the end product, so we don't waste the enzyme here. So we are optimizing the condition. So then how the degree of hydrolysis varies with the different concentration. So degree of hydrolysis 28% at that time. So you can see the characterization chart here. So you can ask one question. Because bromelain and papain we use as a case study in the two system, soya and soya protein here. So other enzymes also has been used. So you can see that how the degree of hydrolysis changes with different enzymes. So another thing is that other source of enzyme like the microbial source and any other source can be used. It's just to clarify the fact that this cannot we can't use in other source in other format. So there is flexibility in that. The science exclusion chromatograph says that the reduction means the idea of cleaving the peptide bond means we are reducing the molecular weight size. We can monitor with simple experiment with is a electrophoresis experiment. So glycine and acidic subunit and the basic subunit, they are susceptible to the enzyme. When the enzyme is acting in this, depending on the polypeptide will break the peptide bonds and the smaller fragment will release. Means size reduction is taking place continuously. Once the reduction is taking place, you have to monitor how the molecule is being cleaved and how the lowering the molecular weight is happening over time. And so different fractions are being separated using the size expression chromatography. So each fraction can be collected. So this is we're talking about bioanalyzer. This is one of the technologies people usually use. Like it's simple, like you have gel electrophoresis. Nowadays they are putting under densitometry scan. So one you scan, you get the profile. It's similar software is there. When you run the electrophoresis, it will integrate the peak and then give the peak attention time in fluorescence unit. If you fluorescence like you use absorbance unit, it's like something here. So it means both acidic and basic subunits are susceptible to enzyme. So acidic and basic means according to the PI, they have been classified as acidic or basic. So here, different enzyme activity level. I think 6 is. Please look at the number 5. So we are talking this is the acidic subunit, and then the basic subunit is here. So you can see the molecular weight marker is here. So the idea is to showing this. You look at the number 6. So, you can see this is a hydrolyzed protein here, papain hydrolyzed protein here. So you can most of the subunits that has been cleaved and the disturbance the high molecular weight protein has gone. So then there is crowding of the low molecular weight protein. So you can say the enzyme is acting in the substrate. The acidic and basic molecule has been broken down. In turn, smaller molecular weight polypeptide has formed. So you will see at the length 6, they are increasing, and the bigger molecule is reducing. That's also corresponds to the size expression chromatography. So we're talking about heated versus non-heated. Heated versus non-heated means if you have we're heating the protein, we thought we can denature the protein. The DS experiment confirms when you heated about the denaturation temperature. in the cooking procedure, protein is denatured completely. So no longer, so I'll talk about the configuration of molecule. What is the most stable conformation? Please tell me, if you put in a genetic configuration, what is the most stable structure? Okay, I'll tell you. What does that configuration? Most stable structure is alpha structure. Okay. Then followed by beta C. Random coil is the most denatured protein will have random coil. You take it. So alpha helix is the secondary structure. The secondary structure of alpha helix is the most stable structure, and beta C is the most stable structure here. And we will be talking about alpha helix, beta C when you will be characterizing the protein molecule by using either Fourier transform infrared spectroscopy or FT Raman transfer Raman spectroscopy. So they have these are vibrational technology, and due to the dipole moment, so that their configuration of protein, the peak will be appearing and disappearing according to the structure. So suppose you have a very expensive protein. It's a function of enzyme. So this, then I'll be discussing about how to design dilute minute amount of solution where circular dihydrogen can be CD experiment can be used to determine the protein conformation. Because some cases we need protein in less quantity, some we can also afford to have in grams quantity, some we can only afford to have in microgram quantity. So if the protein is soluble, highly soluble, and very expensive protein, you want to measure the secondary structure, want to know the structure, so I will advise go to the circular dihydrogen experiment. And if you can afford this in grams, it's better to go to vibrational spectroscopy, either FT-Raman or in case of GIAN. Why these techniques are parallelly, they are conventional, not conventional technology, but routinely used depending upon the hard and fast rule. You have to make a choice, like different purification protocol. What is best for you? You have to decide there. So that way the structural determination happens. So soy protein hydrolyzer, if you look at that, please look line number 5 and 6. This is the control protein here. I can link the hydrolyzed protein here. You can see almost all hydrolyzed. Means the disturbance of brand. So this is a very simple experiment where we can monitor ourselves. Once we hydrolyze the protein, suppose you have different time codes, 5 minutes, 10 minutes, 20 minutes. You can just load those into the lane and you can observe the very good nice pattern the protein is disappearing. And that corresponding to your size exclusion chromatography, different fractions. So we are cross-checking whether exactly the protein molecule is breaking down or not. So what happens when you all know the isolated point due to that ion zero there? So what happens to the protein in case of the time of hydrolysis I'm talking about in hour? So this is 7S, this is 11S. 7S is the beta glycine, and 11S is the major storage protein, that is the legumine or the globulin. So the glycine, different terminology being used. So you can see at 9 hours, just even though this is susceptible to proteolytic activity, still the protein is being cleaved until 9 hours. You can see the major bands are disappeared. So means that's why I told that these are kinetically driven process. So enzyme is acting on the substrate, and the substrate is the storage protein being cleaved over time. So hydrolysis is being produced. So time of hydrolysis also plays a significant role in the production of the end product. So we have to characterize each peptide, each molecule, the fragment, then correlate back, and then say we need this fraction per hour of scale. What's the fraction? It's the same way. That's one protein in industrial scale. So if you can apply to that product. So what happened? We talked about different buffer environment. Which pH is one of the classic examples. If you change the pH of any protein, so we know that the base has dropped in the solubility. So when we come into that, the solubility of protein is near isolectric point is different. However, from GIAN, net charge induces then the steric hindrance will be there, so that the conformation will be different. So you can see the protein is due to the pI of the protein at this range. So the protein is at the susceptible to that. So you increase this speed stillness. So what it means? Means the protein molecule, when we talk about the effect of pH and the structure, secondary structure, we'll see that protein denaturation takes place at acidic, extremely acidic pH and at the extremely alkaline pH. If you add sodium hydroxide, which is 1 molar of an RPL protein, so protein will completely denature. Okay, that's classic example. You add hydrochloric acid to protein, protein will denature. That's why the acid, we talk about there's hydrolysis being producing acid or alkaline. We discuss, then we come to enzyme. Initially, we discuss about different ways of producing hydrolysis. So this is the example. So we discuss why you are not going to use acid hydrolysis or why you are not going to use alkaline way of hydrolysis. So you can see extreme point the protein has been acidic and then hydrolyzed. Some protein may not hydrolyze in alkaline. Yes, so this is susceptible to that. So then effect of temperature at similar pH, neutral pH. So if we studied denaturation temperature, which will increase. Protein at one pH means one configuration, it's a neutral pH, and then we are increasing constantly temperature. So we are going to the, we discussed about onset temperature, then denaturation temperature. So in that way, so you are unfolding the protein, then we are talking about protein is being denatured. You can see increasing the temperature, the molecules are 80 degrees. You can see the higher molecular weight at this chapter. Means the temperature has also effect. When we have been talking about effect of pH, temperature, then pressure. So I'm not talking about pressure here. People have produced product using high pressure processing. It's another area. So
this temperature is the temperature at which hydrolysis is carried out.
yeah, this is the range of temperature. We use 50 and 60. So what happens with another parameter is, can we see the hydrolysis offering different range of enzyme or not? So only talk about papain here. You can see the molecule is being cleaved completely. And what happens is at this, so it means you can produce some of the peptides depending upon your experimental design. You can have control, 100% control. Acid hydrolysis, you do not have that much control. And you are introducing some of the artifacts to the product, the detrimental proteins. In the industrial relevance, everything has to be correlated to the bioavailability of some of the essential amino acids. So mostly we know why we drink milk before sleep. It's a very simple question. In the evening, just before sleep we drink sleep. Recommended. This is why. Yes, that's correct. Because the bioavailability of tryptophan will induce sleep. It's a precursor for sleep. So, upregulate hormone, then we get sleep, fall asleep. So, you get nocturnal milk. Nocturnal milk in Germany, group of scientists are working on nocturnal milk. Means when the cow comes back in the evening, they only produce those peptides, those amino acids rich in the milk. The calf will eat those milk, drink those milk, and then fall asleep quickly. And they got up with when the sun comes in the morning, then they get agile and active. You compare milk collected in the morning, milk collected in the evening, characterize it completely two different things. What a wonder. Same thing. So the idea is nature and everything. So we have to follow actually we are going to follow the same model here, the natural way. So then we are coming about lysine, the methionine, tryptophan, tyrosine, say leucine. Those are the essential amino acids. FO has classified, and deficient of lysine, limiting essential amino acid lysine. So any food is deficient in essential amino acid means we have to fortify it or we are dependent on another source. So complete food means milk is a complete food. We all know that milk is a complete food. It has all balanced nutrients present in that. So compare the soybean with other protein, oils and other. Routinely we are taking that. So you can see this is the amino acid profile. I was talking about the bell shaped top. I think I have given in the tutorial how to measure the solubility. This is a standard format you can get from any of the protocol from anywhere. But for our benefit, I put something. So this is the near the isoelectric point I was talking about of the soy protein. Higher isoelectric point, the solubility increases. So we have to know this principle where the protein is being highly soluble. So that way we have the access to the bioavailability will increase, functionality will increase. Otherwise it will precipitate out, it doesn't get homogeneous system. So mechanism of, another is the mechanism of fat holding capacity of hydrolyzing proteins. So fat binding, fat holding, emulsification, gelation, aggregation, these are the basic terminology I will be using because in food, these are normally used everywhere. So when it is a functionality, so these are the main functions. Then, how to characterize those things? What the principle is happening? You can see the fat body molecule interaction taking place with the entire. This is happening before the hydrolysis. You have same native protein. Then you do the enzymatic hydrolysis. Then the environment of the molecule, you can see they have the hydrophobic regions are available. That's why the ionizable and they are ionized, and then hydrophilic groups are exposed. Means by doing hydrolysis, you are exposing the hydrophobic groups, and you are making the area more available. So the binding takes place. It's a simple formula. Spread a cloth, and then when the active sites are exposed, automatically the things will come and bind it. And you fold it, and then make like a round set something. The active sites are not exposed; they are buried inside. The molecule is not going to interact. It's the same thing happening here. So native protein, hydrolyzed protein, then determine factor. We're talking about soluble, non-soluble, insoluble, and the idea is the native protein. If you go to the hydrolysis, they interact with soluble protein and then the hydrolyzed protein. So the molecular reduction is happening here. If you look at the interfacial stabilization, it's a complex graph, I agree. But the mechanism has to be there in the clarity in comparison to that. So if you look I was talking about more of the hydrophobic groups are exposed when we expose the hydrophobic groups, means you can have more active sites, then the protein is more accessible easily. So the idea is that it's just a representation of that. Any questions here?
The exposure of hydrophobic proteins will increase the binding of the fat-rich molecules, not the active site. Active site will be distorted. When we open, we leave the hydrophobic and hydrophilic groups. So the active site will be distorted. there will be no active site, but there will be a site with hydrophobic amino acids, which will do interaction with fat molecule.
Yeah.
So the previous slide. So that statement explained the enzymatic hydrolysis can improve efficiency by making hydrophobic agents more available, but decrease fat holding capacity by releasing ionizable.
Okay. I will ask, when you protonated, what happened to the pH? Ionizable group will change the pH. That will reduce the fat holding capacity. If you look into the basic chemistry principle, how the buffering of molecule takes place, when there is pH acidic condition, definitely that will happen. That's the reason, conclusion here. Okay, any more questions? Going through. Okay, yeah. We're talking about the hydrophobicity in kindling, and then how the fat binding is increased by the peptides being cleaved, so you are having the more hydrophobic amino acids are coming. So that way we are increasing. This is just a schematic representation of molecules being exposed. That's a very simple way. So how this phenomena can be correlated with the functionality. What is happening if you expose the protein, the hydrophobic sites are exposed. So that then your fat binding capacity will increase. The mechanism. In gelation, what's happening means they form the junction zones. Junction zones means the molecule has to interact each way. If there's a coagulation, then aggregation, then gelation. Then gelation, there will be hydrogen bonding taking place. So protein molecule, the strong gel, weaker gel have this bonding is not, and what stabilizes these junction zones depends upon different parameters.
In this slide, when we are talking about affinity toward the interface, and the first one is very much clear, but regarding the absorption to the interface. So, what interface we are talking about? It is hydrophobic, hydrophilic emulsion.
If you look at emulsion, milk is a very good example of an emulsion. If you put oil, water emulsion, it has fat. So basically, the configuration of emulsion to stabilize that, to prevent the steric hindrance, we put whey protein, basically then you use at the interface. So the plus will be inside to the negative. That's the idea. It's like a battery, you have plus sign, negative sign. So there is the same thing like the molecules have that way, and the interface is stabilized to have prevent the steric hindrance. So that they are not coalesce. This population doesn't happen. So the molecules tends to react in Brownian moment. So then in any direction they will go, then the molecule will coalesce. These molecules, the fat molecules are different sizes. I will advise everybody of you take oil. This one Dr. Chaudhary has asked is take milk, put under your microscope. It may be polarized light microscope, and then under polarized light microscope, use some of the two different filters and observe what's happening to the milk. There will be distribution of different size range of fat globulins, starting from different nanometer range. So you can see if you leave it some time, they'll coalesce. The fat molecules. Then the challenge for the industry to have a consistent product is to control the size of the fat molecule. So we applied. When I was working with CSIRO, so we looked into applied the ultrasonic treatments. It's a modern way of doing the. We applied pulse electric field ultrasonic treatment to reduce the size of the fat molecules here, then control the actual size range. So it's a flow meter going through. Then at the end, we get the same fat separation, and also the milk is coming same similar fat molecule size. So that way people control, this is a continuous system in industry, so you have to do that way. So please look at those molecules under the microscope, you'll see there are different grain sizes. Due to the topic limitations, I have not put those slides, but I'm saying well, this is how you can look when you go back and look under the microscopes. Yeah.
Regarding here that you have mentioned affinity toward interface, and you have put in terms of decreasing order of the affinity. So we have started A4, A3, A2, A1, A0. So looking at this slide, because you are putting the interface, the basically separation phase of polar and non-polar, if I can assume. So looking at that, can you briefly tell that how this particular picture, that the interface, because we are having one red color and second is the blue color. So what is the actual meaning of this red and blue? Because it will help us to understand how we can, how it has been calculated.
Red is the more hydrophobic peptide, and the blue color is more hydrophilic peptide. Water logging and water hating.
Blue is more hydrophilic and hydrophobic.
Hydrophilic is the blue. Hydrophilic.
So it means that hydrophobic also has certain regions of polar molecules, but the majority of them are non-polar.
Yeah.
While in case of hydrophilic, majority of the molecules are polar and few of them are non-polar.
Are you moving to the next? How we are
We have
doing?
another 20 minutes.
Okay.
Yeah.
We can discuss
30 minutes.
We can discuss more about this. So please keep referring to your book or any question you can read it.
First, I should introduce today's faculty in charge. Dr. Sanjay Kumar, please come. Today's session, faculty in charge is Dr. Sanjay Kumar. Please come, sir. He is the faculty in charge for today's session. So, during interaction, during discussions, if you have any queries, post discussion also, you can contact him. I can introduce myself.
Thank you. Actually, I am Dr. Sanjay Kumar from the Department of Microbiology, and today I have been given the task of coordinating this program. My suggestion will be, first I would like to give you one suggestion about this program. That is, generally when we do this discussion in the panel discussion, often there is a time when we don't have questions from people. It will be better that if we have some questions in return, the list of questions, and they can be discussed and, in a panel discussion, so that we know that how much time we are going to take for the discussion, and we never run out of the questions, because it's very difficult to actually to meet those time limits, and when we don't have anything to discuss. So I think today I will be giving you, especially those MSc students who are here, who actually listen to the speaker, but I never saw them asking questions. They definitely have the question, but they don't ask them those questions. So if you cannot ask, you can please give your questions in writing. So I will give you the voice to your questions. So that is one suggestion from my side. Thank you very much.
Yeah, it's a very good suggestion. We should have collaborative and constructive.
Okay, the other day I said I love my students because for them I'm here. I love the student, my professors love me. So this is the two-way process.
You can just switch on that mic. We can also do.
Well, this is page number 153. The escape phase patterns of the hydrolysates prepared with the main at pH level at various reaction temperatures.
Very
What was the question? question, please. Could you please don't mind. 153.
If we look at that, we have seen that lane 6 and lane 7, that's 70 degrees centigrade, 80 degrees centigrade.
Yeah.
I think at that temperatures, the enzymes won't really active enough to undergo hydrolysis, to carry out the hydrolysis reaction. So still you are able to get in the hydrolysis bands. So far away from the optimum temperature.
Okay. It's a very good question. Enzymes are susceptible to temperature inactivation orders. And then the enzyme, since the enzyme kinetics is driven by the temperature, some of the enzymes still accessing the energy protein which is from the cleavings. That's why we thought about stopping the enzyme reaction in the beginning. When you have achieved the degree of hydrolysis, we stop the enzyme reaction. So this way, even the temperature 80 has gone here, so molecule is still cleaving here. You can see the high molecular weight proteins are still there, but the low molecular weight this protein band is increasing. Means
the reaction is carried out at 70 degrees centigrade, and it is not achieving 70.
yeah,
It means initially itself the reaction setup is made at temperature of 70 degrees centigrade, and any of the enzyme will carry the kinetics.
yeah.
But at that temperature, the enzyme shouldn't go on itself. Had it been it is a gradual process where attaining temperature of 70, then it is okay.
That way we are doing here.
We are attaining
Attain the temperature. It goes to the starts from that's what I'm telling. It's going to increase the temperature. If you say another time goes, 7 holding like getting the 80 degree and holding for different time means you have already killed the enzyme. So there's no reaction. You won't see anything. So the enzymes means when the temperature effect means you have the same enzyme and you are increasing slowly, you are allocating out at a different temperature when the temperature is increasing. Or you are randomly putting some of the samples at different set, like a thumb apart, you set one heating block, you set for 80 degree, and that time you take allocate out. So 2 ways of doing that experiment.
I have a question.
Yeah.
When you talk about ratio of hydrolysis, so what are the factors that influence the ratio of hydrolysis? When we are doing an experiment.
Okay. So we discussed several factors. So particularly concentration we saw 5% and 10%. That's one of the most important factors. We said 10 doesn't have any impact on that. In industry, they are concerned about the cost effectiveness of the technology. Even though you are telling 10%, you thought that might increase the production, but it's not really happening. So you go back. Then otherwise, the amenities of that, increasing higher concentration of enzyme. Also, you are adding some of the unwanted product, and debulking is going to take another loss. So concentration is the main factor here. Then what temperature? Like body 37 degree, you know that temperature, everything is occurring. So what temperature next is? Then pH. We saw the pH. That's why we talk about micro environment. Totally calling this is a micro environment. Then pH, temperature, and then and ionic strength. If you are putting salt in that, cofactor. So those are the factors. But 3 factors are very important. This time, concentration, temperature, and pH.
In case of hydrolysis, we talked about enzyme hydrolysis. And in case of example, acid and alcohol based hydrolysis. So have you tried using both of the methods, acid or enzymes together?
Hydrolysis, acid and enzyme together.
Yes.
Okay. No, because acid will denature your enzyme. So the enzyme is susceptible to acid. If you have a harsh acid condition, enzyme is no longer going to be active in that. You are in starting material, you are denaturing completely your enzyme. So even though you have 5% enzyme, it will not give rise to the product you are looking for. Okay, this time Professor Shukla second time requested me to Professor Shukla second time requested me this morning was said to share some thought about entrepreneurship, the process and all the things. Since we have some time to go through it, nowadays everybody is interested to take that idea and then go to the market. So that's called entrepreneurship. But your idea should be viable. You can sell to somebody else. No, it should be unique because you are going to compete with somebody else. It's a very competitive world. First, we should have a business model. Strategize, please write down. You visit Strategize website, you'll find a business model there. They are the best in the world. You can generate business model. You can Google your university. Each Berkeley has their own business model. You can find a template. Means everything has been summarized in one page. So you can write your products, services, goals, pain, gain, everything in there you will write. So you should have an active business model. What you are going to market means you have designed, developed, tested, and produced some novel product. Use the word novel product. It should be new. Okay, otherwise, there's something crowded. Even the product is good, your sales traction may not be there. Something unique, positioning product, you serve. Then these are always consumer-based product. Means you should have consumer insight also, because the key driver for Indian consumer to the other world, part of the world consumer completely different. Even though we are staying in two different countries, so that some will be the overlapping. From my experience and my study and the way I position the product, if it is Indian market, first key driver is price. Okay. Second is quantity, not quality. Do not mind, this is a fact. Third, product application, quality. Fourth is health, health is unreal. So, if you look at the other side, first is quality, your health. Okay. Second, product application. Suppose I want to buy this. I give example. I want to make curry. I want to make lassi. I want to make my yogurt dipping like that. I want to make my raita in that. I want to make yogurt drink in that. Whether one cup of yogurt, I can make 10 glass of that. And some yogurt in the market can only make two glass. That is the product difference there, application. Because your texture, flavor, aroma, everything comes there. So see how this segmented in consumer preference. So you know inside consumer insight. So then only you design the product. So then you have to own the IP. You have identity. So nobody is going to copy you. If somebody is copying you, so you have protected the idea. So you all know what happens in this world. Then it can be marketable. The product, it goes to testing. The product, in one slide, so I just think of describing how this happens. So first is you have a business model, then you should have some product to market, it should be unique, you test it, then you develop, design and test. Means, suppose it's food, accredited lab is required. In Australia, nothing is acceptable unless until you do in a GIAN approved lab. So certain rules are operating in those areas. Nobody is allowed to visit the lab, everybody is certified. Then you produce a product, they sign in that dotted line, this product is designed, developed, tested. Then you file a patent, you can get a patent, or you can, this is your graph and design, so you got the product. Then one the product is there, testing. Testing can be different way. Suppose texture, you have to use texture analysis to test this. Viscosity, you measure the dosorimeter. So this testing has to be done. Then microbial testing. Very important is safety. Consumers should not fall sick. Otherwise, they will complain. There will be product recall, factory seal. So safety is priority here. Do not compromise. Zero tolerance in safety. No compromise. No bug inside it. Everything is within the limit. So then, when the product is ready, then there's the consumer testing. That's called sensory analysis. You have the prototype. You are designing the pilot plan scale from the lab, from the bench to the pilot plan. From the pilot plan, you have trial run. If in one go you may not reach there, but there is 6, 7, 8, 10 times you have to run this thing. Then you come to that pilot plan. Then you design the product. When the pilot product is okay, you are happy with the parameter, you have done the testing, you meet all the criteria, and the one go you may not get, but 90%, 60%, 10%. Because consumer is the key here. When we do the sensory analysis, they will give the feedback. Okay, we think that we have done 100%, but you don't think that you have done 90%, whatever 100% you have given, you have done, because your experimental proof and everything 100% you have done. Your experiment and instrument everything 100%. But you don't know that 100% maybe 50% for the consumers, or maybe 0 for the consumer. They don't like it, they'll put 0 there. I don't like it, it's off flavor, 0. So then you go to consumer. Then in a consumer sensory analysis in sensory science, that maybe do a hedonic sensory test, means blindfolded sensory analysis. You compare your product with some of the leading brand of the product with that cohort. Means suppose I have to position for a product for Asian consumer specifically. Sorry, I am categorizing it, but products are generally categorized. Okay, I do not use that word, but you have to take up that product, then challenge with the blindfolded test. They will rank your product, flavor, aroma, texture, color, everything there. They rank is that in a hedonic scale, like online dislike 1 to 9 score. You got the raw data, then you analyze, maybe 30%, 40%. This is from the sensory. There are two kinds of sensory panelists are involved. One is trained, the other is on-trained. Trained panelists will have expertise in that area. Like we have some expertise there, they're trained in one, like coffee or tea, they see it immediately, they say what is the price paid. Because that trained panel. That way coffee board is deciding the price of coffee. We all know higher order rice is very expensive, like basmati, low GI. So this consumer panel will decide trained and untrained. Then trained panel should also match with the sensory data of the untrained panel. Then you get analyze the data. If sensory has given some feedback where color is an issue or any of the texture, then you revisit and again reformulate what's wrong with the product formulation. Then design the product again, go through the sensory. Once the product is okay, it has compared with the established brand, they will rank, I'm competing the top product, my product is 1, 2 or 3 or anything. Select only 3 or 4 products. You don't have to select 20 products. So that should be wide selection. And that should be similar. Thank you. Suppose you have Greek style yogurt, which is highly thick and viscous, and of course creamy. You should not compare with a low fat yogurt. With the two extreme, comparing the same thing, but two extreme points. The ingredient formulation is completely different. Orange cannot be compared with apple, but they are fruit. So it's similar. So that way you compare. Then once you got the data, then reformulate, then redesign the product again. Go similar thing you repeat. One product is there, so you now are in the market. Then second thing is the value proposition. What is the value proposition means? How you are adding the value to this to the product. When you setting the product, suppose I will say that this product is more healthy. It's a value proposition, but I cannot write in the code in the level because the code does not allow me to write that thing in the book. Because it's a hard healthy, I cannot write. But I will put in the electronic media, print media. My product has been designed which will lower the cholesterol, but I cannot write. Consumer has to read that. So there advertising comes, marketing and other things come. Okay. So you have to do prior to launching, you have proper marketing, advertising, sales, everything has to be ready. So one is the business model, second is the business plan. Inside the business plan, you have each product has a set product life cycle. Then you should have like break-even analysis. Means if this product I manufacture with this, you have to do a proper costing here. You can do yourself, or you can have some of the accountant in your team who can do the costing for you. Like you have to have, this is my raw material cost, this is my processing team cost, this is my overhead, and this is my utilities like electricity, gas, and other thing, water. Then transportation, logistics, and warehouse, cold storage, everything distribution panel, and then stacking. Then to operate that, I need website, I need also telephone, internet. All these things have been together in the business model, the business plan. Everything has been taken place. Then you have got margin. What percentage margin? Usually people take 20 to 30% margin. It's nothing written in stone, but that's the value they play 30% margin. Then 30% margin means you can sacrifice to compete with somebody established brand. Say, my product is a better product, but I'm giving it a lower price. Because you are sacrificing some of the profit. Then you will be in the market. That is called strategy. So you develop a strategy. So you can have 25% profit instead of 30%, or maybe 20% profit. So you are getting a road to the market. Then because you have confidence in your product, and pricing is the key driver when people will think twice, same product, different price, and let me try. They're not going to buy here, they'll try, let me try. They'll pick. Maybe they'll pick the best one, they'll also this one. Or by over the mouth somebody, oh, I saw that one. How is it? They don't ask to try it. They'll say, they'll not buy it. Okay, they'll try. Once they try, they'll pick your product or whatever other product. So that way you are in the market. So the next comes when the value proposition is there. The biggest challenge is the investment. The financial aspect, where the investment come from. If any governmental, non-governmental, venture capitalists, banking, personal, loan, anything, so put together. So how? So you have to work this thing. Then coming to one important question is the cash flow. Very first step, the monthly cash flow. So most of your money investment comes from is being invested on just running this to this product, to the market, to seeing the market. Might have taken long journey to that. So you're only spending. So your money is not coming to your account. You have to have patience. So that way, you should have cash flow. Means past 5 months, I have something like this. If you invested 1 million, the second month something will come, third month something will come, in fourth month something will come. It goes like that. So that way the investment, the cash flow comes. You can have the bank will see the cash flow is coming that way. So if you have investor, private investor, angel investor, there are two model works here. Either they can be part of your company, then you share the profits. Usually, sharing the IP, you have developed. It is I'm talking two models about here. If you have designed, tested, developed, you have spent a lot of money and time in doing this. So you own the IP. So you share the IP because he is giving the partnerships, giving funding only. Because you have done all the work. So it can be around 2 to 8% just to get the royalty here. It's called royalty. So profit sharing is then only giving the investment, they takes 10 to 15, 18% maximum. Same thing everywhere. They take investors. If that's a co-funding, so you can have partnership agreement. So how it works? If you are busy, you have to develop, you have to test it and design product confidence. Then going to the manufacturing, whether you are going to set where to manufacture that, I need a plan. The capital investment will be intensely known, depending on the production coming out. Then you look for somebody who has the capabilities are underutilized. So you use contract manufacturing. The word is called contract manufacturing. You talk to them, negotiate with them, build an idea. I have a product. This product has been tested, designed, tested. So they will be partners together. So you utilize the facility, the production line. They won't get your product, you want their product, you work together. Before that, there are 3 steps involved. First is called the agreement. This is called non-disclosure agreement, or this is confidentiality agreement. Means I will not tell anybody what we are doing together. He will not tell anybody what we are doing together. Before talking in Australia or anywhere, they sign the confidential agreement. So I'm bound to that agreement. It's binding by the Australian law. It's known as ASIC. Australian Security and Investment Commission will sign the agreement. So it's called non-disclosure agreement, or it's confidential agreement. Then we talk openly. Second is called partnership agreement. The partnership agreement means we are partner, profit sharing, time allocation, what products, launching, then everything will be in there. So who will be candidate for duty, how much time, everything is recorded there. So you have agreement signed mutually. Then non-disclosure agreement means somebody wants the IP, the other person will not disclose. But you have developed, since you have developed the product, you are bound to get 2 to 8% royalty as a commission. Each product, not one. Every product will be getting that. So that royalty you can negotiate for 10 years or 2 years, or they can give you. That is the product life cycle. Okay, everything is clear now. The major aspect of this, your stakeholders, your clients, everything happens with relationship. There's nothing works here without relationship. Once you disturb that channel, they are not going to buy your product. Even though you have any good product, any best product, if you don't have good relationship, they are not going to keep it. Everything is happening. So you have a good team, have a relationship, then distribution channel. I talked about supply chain distribution channel. Means cold chain. If it's a cold chain, then you have cold chain distribution, refrigerated tank, refrigerated truck. It goes, then it's stored in warehouse, you can access and distribute. There's multiple cities, you have distributor arranged there. They take the percentage, then they distribute. So that way it goes through. It's clear that one is the manufacturing, second is the wholesaler or distributor, third is the reseller, then fourth goes to company. That's the channel.
And one is trust. Trust about the brand.
Brand, yeah.
Yes. Because trust is very important. Yeah. Once it's lost, then people they will not buy.
They will not buy. Once you lose the trust first time, or something wrong.
This happened with me. I just want to share you. I was used to suppose buy this milk, Vita milk from the vendors, local vendors. One day I just got some report from some GIAN, it is true or wrong. Yeah, so they showed that they are using a lot of chemicals and all this. The day I saw that report, after that I never bought Vita.
Same with
Yeah, I never bought. Amul I have not read yet. If Amul, I will read this, it happens immediately. The trust is lost and then we are suspicious about it. They are telling that they have recovered this problem, this, that, but definitely at least people like me, I'm not able to buy. Even one day, sir, there was no milk at my home. I went, Amul milk finished and Vita milk they were around. I didn't buy. So this is very important, trust. And trust, if it is lost, then it's lost. It's never regained. So I think Sanjay, you can finish the session. Thank you.
If there is no more training, then we can.
I think it doesn't move.
Thank you so much, Dr. Rao. So with this, we come to the end of the first lecture. Now we'll be having a tea break for 30 minutes, and we'll assemble here at 11:30 for lecture 2.